Now, I talked about catnap, and that's we always want clever acronyms. So this is to compile, analyze, and tally neutralizing antibody panels. Okay. The point is that for starting about 10 years ago, people started to be able to go into the sera of chronically infected HIV patients and actually pull out broadly neutralizing antibodies. And that's the holy grail for us, is antibodies that will neutralize a broad spectrum of HIV variants. And not only that, you have to be able to go and prospect and actually pull out not just the antibody from the serum, but the B cell from the PBMCs, and actually go in and pull out those particular B cells that produced particularly broadly neutralizing antibodies. And that's great. But like every research advance, it has a downside, which is all of a sudden there are hundreds and then thousands of antibodies being tested against hundreds and thousands of different viruses, which makes it for a data nightmare. And so the question is, for everyone in informatics, generally speaking, is how do you pull out meaning from data? And in this case, this is one approach where we've built, and by we I mean Karina mostly, and HIV Immune, and other people, not me. It's a scientific we, which means them. So what they have done, our group has done, is put together a combination database access tool, and what it gets you is the binding region, the neutralization for each individual strain, each individual viral strain by each individual antibody, and the relevant sites, contacts, positions of interest, and so on. And it's a complicated tool with a bunch of ways in, and so what I'm going to do is run you through it in live demo. So let's see if we can make this work. Good. So I'm going to page.lino.gov, immunology database, tools, and so you see what I just did. This is just kind of an interface thing. So clicking on the tools gets you both the drop down, where I could go to catnap, or if I click on it, it gets me the tools page, where I can go to catnap, which is what I'm going to do. So there are several variations here, and one of them, for instance, is straightforward catnap. Next is custom input, where you can put in your own data, and this is kind of groovy because you can put in essentially any protein sequence and any quantitative data associated with it, and do prospecting for which sites are important. And finally, the hybrid is you can say, well, I've got my HIV sequences and neutralization data. How does it compare to what's already present in the database? For now, I'm just going to go to the straight up catnap, and let's see. And I'm going to run you through this at some length. Feel free to stop me, and when you don't understand what I've just done, give me a shout. Okay. So what we have on the left is a list of antibodies, and as you can see, it's a fairly extensive list. These are all monoclonals. And over here we have a list of associated viruses. This is not all the viruses in the database. This is a selection of viruses all of which have been, for which we have neutralization data for one of these, for at least one of these antibodies. And there are quite a few. So what I'm going to do is pick. You can, of course, select everything and do an all-by-all comparison. but that's a little sluggish for this purpose. I'm going to select a handful of antibodies, 10, 1074, and I'm going to do, I'll do 10E8, which you saw previously, and I'd like to do PG9 as well, but it's a pain to find it here, so I'm going to do a command F to get me get down to where that is in this list. So I've selected some antibodies, and I can say, give me details about those antibodies. I click submit, and sometimes people say, well, I clicked submit and nothing happens, and that's because the data comes out at the bottom. So scrolling down, you can see that we've got the antibody name, we've got where it binds, we have links to PDB showing for the structures. Hooray! And then there we go. So that's helpful. And links to donor information and to the paper from which it came, and for the antibody features that Elizabeth showed you a minute ago, as well as various predictive sites. And importantly, there's also the germ lines for this particular that were associated with this particular antibody. But wait, there's more. If you click expand, you also get the sequences of the antibody from the heavy chain gene, light chain CDR3 sequences, as well as the whole darn thing, and aliases. That is to say, other names by which this antibody is known. Is that a question or a stretch? It was a stretch. Okay, no, it's okay. Stretches are good, by the way, because otherwise the blood pools in the extremities, and then you're in trouble. Okay, but this is actually, sadly, not a trivial point. Is that you may have this antibody 10-1074, or wait, no, it's 10.1074. Oh wait, no, it's just 10.1074 with no punctuation. This is the sort of thing that makes the informatician's life miserable. And we have, again, we, not me, spent a lot of effort making sure that we have all the available aliases for all these different antibodies linked together, so that you get what you want. I will note just as an aside, this sort of thing makes me very, very sad, because this is the antibody 10E8, which has been interpreted by some software we won't name, Microsoft, to be a numerical value. That is to say, 10 times 10 to the 8th, or 100 million, sorry, what's 1 billion as it were. Come on, guys. But I digress. No, I have a whole series of a study with monkey names, right, where one of the monkeys was named, unfortunately, 10E96, which is a great big number. And let's not even talk about dates. Because poor monkey, August 24th. No, that's a sad thing. But anyway, we have a ton of antibody data available here. But we also, if you scrolling, okay. In addition, we can get viruses, and you can select several or all the viruses. For instance, if we just wanted to get the virus details, we click that, and again, oh my goodness, nothing happened. Well, yes, it did. It's down. It's actually just there it is. So virus data, analogous thing, everything you might want to know about these viruses, what they're called, what their subtype is, where they came from, what about the patients, and so on. But again, that's not enough. You might want to, we don't have to expand here. No, sorry. So you can also download the sequences if you want from any particular so that's all great. But you might also oh, the other thing is that there are a lot of these standardized viruses used for neutralizing antibody, used as panels. So in other words, I've got my great new antibody. Well, how good is it really? I don't know. I'll pick a bunch of viruses out of the freezer and see how well it neutralizes them. And people did it in exactly that mode for quite some time. But eventually, it became clear that it might be nice to have something reasonably standard. And so there are standard neutralizing panels that a number of people used. But I say, notice, not a standard panel, but standard panels, because we can't have one standard. We have to have 14 different ones. But I digress. So in any case, you can choose, for instance, I'm going to go back here and select, have all my viruses selected, and I've got my antibodies selected on this list here. And the key point, the most exciting thing here would be is to get the assay details. So I'm going to go ahead and submit this, and I think I will exclude viruses without sequence data and submit that. And now this is sort of where the rubber hits the road for this this tool, because what we have here is first a list of different viruses and their sequences, and a list of the these three antibodies that we asked for, and here's the neutralization values for each of those antibodies against that virus. And so I asked for all the viruses in here. There are 1,000 of them, and we can scroll through, and you notice that these are all synchronized. And notice also that here we've got a red N. The red N is an asparagine. It's in N-linked glycosylation motif, meaning it's likely to be glycosylated, which it turns out is hugely important for HIV as well as other pathogens. Because a glycan is about the size of the business end of an antibody, so it really makes a big difference. As you might expect, we can click to sort, and so if we go, so now we're sorted by the best neutralizing titer, that is, let's say the lowest IC50, and notice that all the viruses have been sorted that same way, so that we can now go through here and look for motifs, generally speaking. And again, you'll note that you can scroll all the way through, and the ideal place to put this would be in the vicinity of the epitope. And I will notice also, depending on your browser, it's frequently possible to resize these windows, but you'll have to play with that. I'm not going to do that live now. And of course, everything is downloadable. So let me scroll down here. And of course, we've got all the papers associated with it. We've got contact positions. We have a wide variety of data here as well. Now I'd like to back up a little bit and show you one other way through here. Are there any questions so far? Is what I'm doing here is perfectly limpid and clear and sensible and well understood? Yes, no, okay, anything. Got some nods, okay, fair enough. Let me just go back straight here to all the way back. Because I'd like to go just show you one more groovy thing here, which is going by study. So there are a lot of these, a lot of papers where people have taken their antibody or a set of antibodies and gone against a set of neutralizing virus panels. I happen to to like Mattia Bonsignore. He's a great guy, and we'll look at this study of his because it's got some cool things on there. But again, you can do any one of those guys. So again, we've got the same panel. I'm going to show you that I can expand here and find out everything I want to know about each of these viruses here. Likewise, I can expand here and look at all the different antibodies used in this study, right? One column per antibody. And I think I can even, yes, I can get more sequence because we've got a big screen. We might as well use it. So I have to be able to find the scroll bar. So I talked about the relevant positions here, and for each of these antibodies, we have determined what's been reported to be the relevant amino acid positions for each antibody. And these are not necessarily contact residues, right? Because it turns out there are in many cases, not just HIV. Of course, the residues in the epitope itself, where the antibody meets the protein, are important. But there also can be residues elsewhere that will allosterically affect the shape of the epitope. And it's just an aside. It's not always about contact. It's important. But I'd like to look at this position N332. Asparagine there, and when I click on it, something ought to happen. Yes, no. But sadly, it's not. Bigger in the hopes that I'm going to get the link. Oh, there we go. So I got caught by that. So when I clicked on this, that ended up filling in this number here. At which point I'm going to click Analyze, and then I think something's going to happen. Excellent. So two things happen there. One is that, and I need to make this smaller again, is that the position 332 is highlighted in the alignment. So that brings me there. That's what I care about. And the other thing I'm going to do is, now I said, right, this was for antibody 10-1074. Let's sort by that. So now we've got the best binders at the top, and I'm going to click again to get the worst binders at the top, and we'll just scroll down here. And the point is that these guys that bind terribly, that have no color here, are mostly not N. And when we get down to things that start to bind a little bit, they're all N. So this is just eyeballing it. It looks like having an asparagine in that position and having a red asparagine particularly at that position, that is red meaning it's likely to be glycosylated, is important for antibody binding. So, but that's all fine. That's qualitative. That's what a biologist would do, right? A biologist would look at that and say, oh well, there's something there. But a statistician would want a p-value, and so well, we have, fortunately, I guess it's fortunate, I think it is, we have some statisticians, and if you look over here, we've done just a simple Fisher test, saying, well, it makes a difference if you have an N there to start with, right? So having an N versus not having an N at that position has a pretty decent p-value. there being an effect there. But also, if you look down here, it's it matters whether it's in a glycosylation motif. So whether it's G plus or G minus. So again, this is sort of telling you, look, glycosylation of an asparagine in that position is really important for this this antibody. And again, we've got tons of features here. So more. I was given a book, quick aside, which was entitled "More Than You Probably Want." No, almost certainly is the title of the book. Almost certainly more than you want to know about the fishes of the Pacific coast. Okay, it's a good title, and it's absolutely accurate. We have here, almost certainly more than you want to know about any particular antibody. But if it's the antibody you want to know about, it is maybe what you want. So that's essentially catnap for now as an example of the sort of things that are available. And again, we've got 1,000 virus neutralization data, and I forget how many monoclonal antibodies in there. But this is kind of a cool tool to play around with, and if you have any interest in neutralization of antibodies, it can be helpful for other pathogens as well as HIV. So the slides will show you both a theoretical approximation of a catnap. Now, what's the deal here? I think this is funny. I didn't put it in, but it's a dog taking a nap, which is like a cat nap. Okay, all right. I think we'll take it out because we keep showing this and nobody ever laughs at it. But Peter was involved in this tool, and this is his dog. But in addition to the cute picture of the dog, you can there are examples that you can follow along here at your leisure, as I'm sure you have it. I've shown you most of this stuff. I didn't click through. There's information about a particular fella who generated this antibody. Unlike many people who generate broadly neutralizing antibodies, this one seemed to help him. He was doing pretty well long term. In most cases, people who develop broadly neutralizing antibodies actually are not are actually pretty sick. So again, I've gone through most of this stuff live. This is just sort of a guide to show you where to help you do this. I'll point out you can get to the sequence database for the full sequence of all viruses if you like. And I didn't point out the sequence motif here, which is a combination of the residues of interest, stacked up with sequence logos, which you guys have probably seen, showing where the height of each letter is proportional to the frequency in the sample. Okay. So that's catnap, and I would be thrilled if any of you have any particular questions on that. Otherwise, I'll move on to Combinammer.
I'm going to pause you there.
Okay, good.
Yes.
Great time.
Great
Excellent.
Time. Well, sort of great time. We're pausing database. If you're looking for any individual attention with any of your queries, B-cell, T-cell stuff, we do have 2 rooms set aside, and so you can get some one-on-one individual attention on that. Otherwise, just hang out here, and we're going to continue with the HIV stuff, and then we will all reconvene or meet up again on the back patio at 4:45, 5 o'clock to do our reception. So we'll give you guys a couple minutes. Yeah, of course. I'll give everybody a couple minutes to get your stuff together. If you do want to go to a separate room, you can follow these guys who will be going to those rooms. And for you guys, we have T-cell in T2101 and T-cell in 1103. You can lead the charge, that would be fantastic. T-cell 2101.
we will have tomorrow also another individual session after the talks in the afternoon. So if you have any questions for T cell, the room is 2101. It's on the second floor. So you can follow us or join there later.
I would say
something bad.
I want to ask you about protection. Because I couldn't
All right, and then we're going to go ahead and get started just so that we stay on time a little bit, just a little bit. So goal for your reference is to end by 4:45. We've got 55 minutes.
We can't do that.
All right.
Well, thank you for your gluteal fortitude in remaining here and seated. Combinator is another cool tool that we made, and by we I mean mostly Shinich and Shinich embedding, not me at all. But the point is, again, neutralizing antibodies, hugely important, but as I may have mentioned once or twice before, escape is a problem in HIV, and antibody escape is no different, really, from drug escape. And there's been some recent studies showing that, as we told them all the way along, monotherapy, even with a great broadly neutralizing antibody, results in the development of rapid escape in almost all cases. The virus just walks away from it. So by analogy to multi-drug therapy, which is very effective because you force the virus to make multiple mutations at different sites and get them all together in the same genome at the same time, which becomes statistically difficult for it. By analogy, as I say, to multi-drug therapy, multi-antibody therapy could be fine, with the caveats that delivering a multi-mAb cocktail to 30 million infected people is going to require several tons of monoclonal antibodies at the price of hundreds of dollars per microgram. So you do that. But nevertheless, an important research question. And don't get me wrong, there are ways that this could be made to work, but it's not trivial. But the point is, you might have several antibodies, and you want to know, well, how well are they going to work together? So, given neutralization data, that is, IC50 or IC80, and some antibody targets, you can use some stats. I believe it's Bliss-Hill statistics to make a reasonable prediction of the efficacy of this particular combination of antibodies. And so you start basically with data like this, where you have each one line for each antibody, and essentially as the antibody concentration goes up, the number of viruses that you can neutralize goes up. And ideally, you'd have a very steep curve far to the left, where with where very low concentrations of antibody will neutralize a whole lot of viruses. Obviously, what we have here, even for these very nice antibodies, is not ideal. But the interesting thing is that there are different curves here. For example, this guy starts off strong. It binds some fraction of the viruses very tightly and neutralizes very well, but it tops out. It misses quite a large fraction of the tested viruses. So you can combine them in two cocktail combinations, and for example, if you put the CAT256 antibody, which is light blue here, together with 1074, which we talked about here, which is green here. You combine the blue and the green here, you get this red guy, which is doing all right because they bind at different sites. Likewise, you can add a third one in there, and you're doing better still. Adding these four together are actually not that much better than the three. So there's a artifact there with the scale. But so the best single antibody, best double, the best 3 and 4 are pretty close. So that tells you, where you should go in terms of designing these studies. So that's common. Any questions on that? And does anyone outside the HIV world care? Yes.
Yeah, so you can go scroll through all combinations from a list of all the singles, doubles, triples, quads.
Let's go.
Oh, that's what actually what happened.
Because it's probably easiest just to show you. So I'm going here to tools, and I'm actually just going to. I can either look through here and find Combinabber. There it is. So I'll show you this. You got this tomorrow too. All of our tools have some sample input. So if I click on that, that shows you what this looks like. And this is sort of terrible, but let's go look at what that does look like. So we have here, so the input is a list of viruses and a list of antibodies and the neutralization for each of those viruses. So there should be a one-to-one correspondence between every virus name in this row and I'm sorry, am I going to do this right? No, I'm sorry, it's antibody names in the, I'm squinting here, in the top row and virus names going down the side here, and the neutralization is in the middle of the data. So just throwing that in as stated will give us, make this a little bigger. We have a variety of options, but I think we're just going to use defaults. And in a matter of moments, we'll have something. Okay, so this is in fact giving us a list of these are the same figures I think we just showed you before. So the single antibodies and the combinations of 2, combinations of 3, and combinations of 4. So that's what you get if you do it with your own data. If you gave it only 3 antibodies, you would only get combinations of 3 because that's just we can only do what we can do, right? We're limited by the realm of possibility. Okay, so that's that, and there's if there's a paper on it which is in the slides, and you can check that out, and if there are questions about it, you can mail Kshitij, because he knows about it, and I don't. So the other thing was, and I think I don't know how this really works, because we're sort of ahead of time. I'll give you a little bit of the tour in the genome browser, just because it's cool. The point of this is essentially there's a lot of stuff going on with the viral proteome, the viral genome, and you might, for example, not actually. Have a particular region that you're interested in. You might generate some sequence. You look at it, you say, oh, something's going on here, and you can go and look at it. And I won't be too discursive here. The point is simply that we have this browser which is built on JBrowse, a tool for large genomes. One of the nice things about working on HIV is it has a little tiny genome, but there are big genome tools available. So we have an advantage there. But basically, you have the map, all the genes of the genome with their stupid little spliced genes, and there are three coding sequences in the same overlapping. All the things that makes HIV bioinformatics such a pain in the neck. And you can add in all the known antibody epitopes that we have, CTL epitopes, and if you go and say, well, there's something going on in this region in my sequences, and then you can go and say, oh, well, look, there are a bunch of antibody epitopes there, might be relevant, and you can zoom and scroll through the whole thing. And I guess I won't go into too much detail, but again, when you zoom right in, you get the actual epitopes that are present, and as well as antibody contacts and other features. So, for example, this is one particular amino acid you zoomed all the way in, and you can say, oh, well, that's important to binding to these antibodies, that particular residue. Again, here are the CTL epitopes and also helper epitopes. So, I think Elizabeth was going to take it from here to do a quick tour through some of these guys, and then I'll come back for the mosaics at the end. Was there a question?
Yeah, I have a question. So in the genome browser interface, do you guys have like transcript label information, like mRNA label information?
I'm sorry, what do you mean by mRNA label?
Sorry. Okay, it's fine.
No, I'm happy to take the question. I just don't know what.
So okay, I think I misunderstood. Sorry. No, I think I kind of understand now. Sorry.
Okay. Well, if you like, I'll why don't we swap off, and I can.
I have a question
Yes.
about your monoclonal antibodies.
Yes.
How do you deal with? I mean, how many unique patient-derived antibodies do you have? Because now when they have the whole families of antibodies derived from one patient over the time of the infection, do you capture all of those separately, or so how many kind of unique origin antibodies do you have?
So in other words, if we have one patient like CH505, for instance, where we've got a whole series of separate antibodies, is that we've got 20, let's say 15 separate monoclonals from that patient?
Yeah.
Is that, are you talking about as one origin?
Well, yeah,
or
or they have it over the course of the infection, so the antibody has changed over the course of the infection.
Yeah, in fact, the interesting thing is on those guys, and I'm not sure if I don't think I'm going to answer your question first, which is the how many question, because I don't know.
Okay.
But the interesting thing is, there are in fact, in one patient, you have different antibodies coming up from different germlines. They're totally different origins that actually may compete with one another. And in fact, there's several cases where escape from one antibody has driven the virus into the arms of another antibody, but because the escape form is from the first is sensitive to the second, which then comes up. And so there's this whole hideously complicated game of cat and mouse where the different viral populations are coming up in response to different antibody populations being driven down. And so
Those are all captured independently as
those are
line items in the database?
They are, yeah. If it's a separate antibody, that is to say, it's we have the sequence and and can infer the germline. Really, it's just those are in there separate. They will be derived as being from the same patient,
Right.
and so you can get you can get those. It's an interesting question though, because we have for the sequence database, for instance, ways of getting only one sequence per patient.
One viral sequence.
One viral sequence per patient. I don't think we have an analogous automated query to get only one antibody from the same patient. Again, for most patients, there is not a large number of antibodies that people care about because most people don't make, because most of these are of interest because of their breadth, and most people don't have multiple BNAbs, multiple broad neutralizing antibodies. But it's an interesting question to think about. Okay, Elizabeth, take it from here.
To answer your question, yes, every germline variant antibody is a separate, and it can be gotten to separate.
Right, and even those that are manipulated
Yes,
in vitro.
sometimes
And-
even
and
manipulated in the
they're
lab.
They're called the same, yet there's a mutation in them. You have to kind of follow.
So if it has a different name,
Sometimes they don't change the name, they just.
yeah, if it's just like a few residues and they haven't changed the name, then that will not be a separate entry. But if the author gives it a .02 different name, then yes, it's a separate entry. But it will be cross-referred in the notes. So you can actually, if you just look for a few more seconds, you can find out where it came from. So we saw some of the more recent tools and things that Will went over, and I'm going to help you through a few more tools that we have that are of special interest to immunologists. And as you can see, when you look at this, you can see that there's these little colored boxes on the left, and the blue ones are tools that are good for general use. So any organism, if you have information on sequence, you can use these tools for any organism. The red ones are just only HIV. So you can see that actually, and this is not all our tools, this is just a few of our tools. But in general, we have more general use tools than HIV specific tools, and the ones that are purple are the ones that are general use, but they have a few HIV specific features. So if your organism of interest is not HIV, which I know it is not, these can still be of use to you. The other thing is, as Will mentioned before, almost every single tool has a sample input, so you can run that and just check out how it works. All right. If we were to go to the HIV sequence database and look under the Tools tab, you can see that you can get to pretty much any tool. There are also tools under the Immunology Database tab that are only for immunology purposes. Well, I shouldn't say only, more for immunology purposes, but they can all be got through from the sequence database. And so that would be something like this. Here you're at the Immunology Database, you look at tools, there's fewer of them, and let's check out some of these. The first one is the HIV Sequence Locator tool. It is specific for HIV, so I'll be very quick with this one. You could put in, if you were searching for a certain epitope of yours and you wanted to see where it falls aligning to the HIV sequence, it will give you that aligned to the reference HXB2 sequence. And you can put in just the epitope that you're looking for, or indeed any bit of DNA. I'm saying DNA, peptide. And you can search for it. The output is in two forms. There's a graphical output here, and you can see that it aligns to GAG protein. And then there's also tabular output, which gives you the coordinates, and then tells you a little bit more about where it falls in relation to the HXB2 genome. And then down here, there's a little alignment, and here you can see its complete identity. Another thing you can do to search on that tool is you could just put in coordinates. If you knew the protein and the coordinates, put them in, and then you get two tables. One of them shows you whether it's completely identical to the HXB2 sequence or not, and the other one puts into the context of surrounding sequence about 50 amino acids surrounding it. Right. Now this is just HIV, but what if you wanted to do something similar but for your organism? we have a tool called PepMap. And PepMap is pretty useful for, especially for making figures and things like that, where you can put in your own query sequence, and then you can map onto it epitopes or functional domains, and you can call it what you want. So for example, here in the input, they've given 2 different peptides. They have an HLA-A2 epitope, they have a B8 epitope, they have a reactive peptide sequence, and they have a not reactive sequence, and you can map them all onto your sequence of query. This is how it would come out to look. Or you can also get a peptide map, and that one you can use to, and you can name different, give it different names, so that makes a nice figure or something more easy for you to follow. And you can also get a table with different, with the same parameters essentially, but in a tabular form. And then there's also a percentage similarity here to your query sequence. That's the next tool. And then the next one that I'll give you is PepGen. Now PepGen is a tool where you can generate peptides to be constructed, say if you have an experiment, off of a query sequence or even in a set of alignments. So for example, if you have an alignment of 3 different sequences and you put that in, and then you say, okay, now generate for me a bunch of peptides that I could order, and you know, or whatever. But generally it's because you want to order them so you can experiment. Then you will actually get, so 2 things you need to put in. You need to put in how long you want those sequences, the generated peptides to be, and also how much you want an overlap of. So if the length is 15 and you have n-1 overlap of 14, essentially you're going to get the whole thing, the whole, represent the whole sequence, but that's not what most people do. It's usually 15 overlapping by 5 to 7 or something like that. So put it in, ask for an output, and then you get 2 different ways of looking at it. This staggered output shows you the peptides from each alignment, each sequence that went into the alignment. So there were 3 there, so you get the peptides from 3, 3, 3, like that. And sometimes there are some that are duplicate. For example, the 3rd sequence here and the 1st one have the same, they're identical. So in that case, this view will give you only 2 peptides because it automatically takes into account that there was 1 identical, you don't have to order it twice. And it will name it so you know it came from sequence 1 and 3. And this column view is useful because it gives them in different blocks. So that's the 1st block, the 2nd block, and these are the 3 from the 1st block. And so it's already pre-named, easy for labbies. Okay, so that was PepGen.
And then just one comment on that, if I might, is that it's not a big savings necessarily in money when you're ordering peptides, not to duplicate one. That's not such a big deal. But if you think about you're going to do tests on these, you know, each peptide is going to require some cells, right? And those samples may be precious. So so every saving in the number of peptides you have is repaid later by the savings in the number of wells you have to do and how many cells you have to consume.
So we go on to the next tool that we have here, and this one is Epitope Location Finder. So if you have an epitope of interest and you want to find out where it lies on, I said that wrong. If you want to find, if you have a sequence of interest and you want to find out what epitopes are there on that sequence, this is the tool for that. And there are 2 things you can come out with 2 results. One is it will find you indeed what are the known epitopes in that, within that HIV sequence. And then the other one is what are predicted epitopes. So the known ones, you can see out here, when you put in your sample sequence, you do also need to have, we don't have to, but you would want to choose which HLA you're looking for. So which HLA's epitopes are you looking for in this sequence? And you can choose more than one. And then another benefit of this tool, which is really great, is that you can actually link to the IEDB epitope. So you can use their immuneepitope.org, the tool that resides there as well. So we have one tool that gives you predicted epitopes, which is Motif Scan. And that's rather simple. It really depends on the anchor residues, but it will scan and give you the different epitopes that might be present and that are predicted within your sequence. But they also, it also links to the more sophisticated tool of IEDBs, which will show you different parameters and different ways of looking at predicted epitopes that might be within a sequence. So let's take a look at that a little bit. The output, if you looked for a certain, if you put in a certain sequence and gave it, say, a certain HLA to look for epitopes, would come out in this form. And you can see here the top three with green arrows are epitopes that are known to be present in HIV, and they are actually restricted by that HLA. And so you can say A6802 restricted epitopes here. There's three different ones because they're small variant. They are variants in sequence, but they are all recognized and they're all presented by A6802. Okay? However, it also gives you other, the same epitope that are restricted by other HLAs. So there's a 68, which is probably 6802. There's 28, and there's 44 here. And it gives you the differences if there are variants in sequence. Okay? Now you can go, from here you can go to a couple other things. You can either click on the epitope and go straight to the epitope records that were curated or annotated in the epitope immunology T-cell immunology database, or you can click to align and see how that epitope, the one that you're interested in, like supposing you're like, what's that? And see how that aligns to the different sequences. And so we're going to see what that looks like. Sorry. We're not going to see what that looks like here because I will later on show you a quick align, which is the tool that is used to get to the alignments. But now let me show you the predicted epitopes and what those two outputs are. So here we go. This is from Motif Scan, which is our tool, and this is what you would see. You would see, okay, we have predicted epitopes, and here they are, and you can see them listed here, okay? However, if you had checked the IEDB box, you go along here to theirs, and if you click on this result, you will go straight to their website, and then you can continue with the links and tools there.
So how good is the agreement between the predictions?
It's perfect. We absolutely agree every single time.
It agrees well,
Okay.
in
No,
general.
it's actually something,
Well, ours-
variable. It's variable.
Yeah,
It really is.
and ours is more, it's just by anchor residues.
Right.
And theirs has other parameters.
Yeah. No, I think, our tools are generally speaking, for epitope prediction are simpler and, on the one hand, and only based on stuff actually reported. We don't do much in the way of we don't do anything in the way of serious prediction. That's what the guys here do.
This is what the IEDB page would look like once you got to it, okay? And then it's gone. Remember I said quick align and I didn't show it to you? Well, this is a different tool, quick align, and it's just a quick way of aligning sequences and then creating different outputs. So it aligns your query sequence to an your query sequence to an alignment. And so if you put in, say, a good old SL9, and you say, okay, give me that information, submit, then you get SL9 and all the different hits that it has to the sequences, and some of them are identical, and some of them are not. There are variants, of course, and this is actually generated by subtype. So A and then B, C, subtype of the virus. Okay? And then they give you a variant frequency summary over here as well, so that you know how often you might find that residue within that epitope. And these results are tabulated as well, if you prefer. And then a more graphical representation, a sequence or web logo, where you can actually see that usually in position 1, there's always a serine, and then, but in position 3, you might have a tyrosine or a phenylalanine. And so this is a nice graphical way of outputting it. And that's quick align. A little bit more complex than quick align is analyze align. Very similar, but you can put in your own query sequence of data. So this is really good for any organism. And then you can ask for, and then you can give other bits of sequence and align them up. Okay? So analyze align has outputs in different forms too. You can choose what you want. It's a histogram at different positions. You can see epitopes aligned to other sequences based on what you put in. There's a tab table, and there's this web or sequence logo, just like the other one. And you can see your residues of interest at different percentages.
We should be clear that
Occurrence.
this is basically a fancy wrapper for Web Logo 3, which was written by somebody whose name I forget, not us, not our tool. But we've got a nice wrapper around it with a bunch of subsidiary information that's A, makes it easier to run Web Logo, and B, gives you a bunch of additional data for any given slice of an alignment.
So the alignment is done on science.
Yeah, the alignment.
You import the alignment already.
Yeah,
done
yeah.
and
So quick align, for instance, is takes a single sequence of yours and pulls out the corresponding slice of our pre-built curated alignments. Analyze align requires your
An
sequence,
alignment
your
cluster,
on
something.
alignments, because the key point is it's analyze on an existing alignment.
So here is another output that you can get. You can get several different views, say across different subtypes. And then you can look at them and say, well, these don't look maybe too different. Some, maybe this one and that one, but the other subtype does. And here's to answer your question earlier. Here's an example from a paper, Pete Praver, the same guy with the dog and the cat now. This is his paper, and here's a figure from it where analyze align was used. And you can say this is a transmitted HIV, and this is it 3 years post-infection. And if you look at it, you can see in different positions, like, for example, the histidine here is very faithful over the last few time points, but was not even, the histidine was it a D or a Y, one of those, before in the transmitted sequence, okay? And then everything that's gray means that it wasn't there before. So there are insertions, and you will also see that sometimes if there's a gray here, there's a deletion by the end. So you can see where there were insertions or deletions. You can see that the relative percent representation of different residues has changed dramatically. And wherever there's an O, it's a location where there could be N-linked glycosylation. So that's also interesting where, and I failed to mention this, but in analyze align and quick align, that shows up as a big pink O, but for this paper, it's not pink. So you can see that analyze align can be used especially in longitudinal studies and when you're following certain epitopes. And so that's a good tool for that. And I think with that, yes?
So read out for these things like ELISpar in like assays, wet live versus ELISpar. So will readout go down over 3 years time from beginning to?
If you were checking on a certain epitope, say there, and it changed, yes, most probably. Yeah, it would change. It would maybe change and go down, or who knows, up. You know, yes, it would probably change. Sometimes the one amino acid difference may not make a difference in the readout, and you'd have to sequence it to know. But in general, yes, it does change the immunological readout.
So the typical kinetics, and it varies a lot from within, you know, and from case to case. But typically, you'll see the CTL response will go up and peak, and then tail off. and it will typically tail off because, once you see escape. And for this figure, they're ordered left to right by the time of replacement of the transmitted variant. So for example, that N all the way at the left, by the third time point.
No?
Yeah, that one. By the third time point, it's been replaced. The asparagin's been replaced by a lysine in more than half the sequences. So that K there is over 50%. So that was the first replacement. It's not, these are not in aligned order. So that's position 279. The next,
Yes.
and it was embedded in an epitope. The next escape to happen, and I think it wasn't escape, was the histidine. It's at position, what's that, 477, 417?
417.
417. So that histidine was replaced substantially later at the. I think it was less than half at about the fifth time point, where there's a combination of H and R of other things that are more than half. So again, that was replaced. That was suffered substantial change early on, and so that's how it goes. And over at the right hand side, that glutamine, that Q, didn't come up until 3 years, almost 3 years post infection.
But it is position 412.
So in this CTLSA, what was your target cells?
Could you say that again?
In CTLSA,
Uh-huh.
what was the target cells? Target. T cells will be like effector cells, which will
Right.
be killing. But what you use as a target?
Well, so in the papers, sometimes
Okay,
it is
it's fine.
PBMC, whatever they did. Yeah. So, and this was just, he didn't actually do the experiments.
I think this is probably one of the chubby patients. I think this is probably
Is it?
CH505, and these are mostly going to be associated with antibody escape. These are probably mostly antibody escape cases, but we'd have to look.
Yeah, I can
There's
check.
the CTL and antibody data for both these guys.
Yeah, I'll check on the paper. Thank you, and now Will take us on to the end.
Okay, so here's another tool, and this again is just this is a wrapper for an R package, basically for doing hierarchical clustering. The point is, generally speaking, is if you have a table with some values in it, you can turn it into a heat map. In this case, it's kind of a cool experiment from one of Karina's papers, where there were a bunch of guinea pigs that were vaccinated by different regimens, and then tested for neutralization against a panel of viruses. So, you vaccinate the guinea pigs, you bleed them, you take the sera, and you put it in neutralization assays. And in this case, each row is a different guinea pig, and the color here gives you the vaccine group, basically. And each column here is a different virus, and the color is I think this is I guess it's ID50. So the highest neutralization is in the most intense color, and so this is just an example figure. You can see that all the guinea pigs neutralize this virus really quite well, and some of them more strongly. And there's a scattering throughout. But the point is, by the clustering that's done in the package to make the heat map, is that generally speaking, this lower cluster, these guys all behave similarly and have fairly poor neutralization. These guys. This group, you see the tree here, have moderate performance, moderate neutralization, and this group all has pretty high performance. And you can see here that most of the animals in here are in the red group, where as it turns out, they had four variants of the vaccine, and most, or in fact all of the red group, are in this higher performing group. So that's simply an example of we had to make some of these figures for work that we did, and we thought, oh, wouldn't it be nice if there were an easy way for people to make such figures? And so we made a tool that would do it. Okay, so I'm going to take us down to the end in the remaining, let me see, 15 minutes or less on these vaccine design tools. And again, this is a question of we have a problem for our research. We have some research questions, and we built some tools to answer those questions, to address those problems, and we put them up on the web to make them generally useful. And this is, as I say, near and dear to my heart, because this is what I came on. This is how I came into HIV, was for this problem. And the problem, as I've said over and over and over again this afternoon, and as I will say over and over and over again tomorrow, is that variability is a big problem for HIV. And the way we addressed this was to generate some, and one of the nice things about working at Los Alamos is there are a lot of smart people up there. The team, quick aside, the team we had working on this project included a couple of informaticians, one or two actual immunologists, a quantum mechanic, right, who's published, most of whose papers are on lattice theory, whatever that is, and a couple of remote sensing guys whose day job is collecting multispectral images from satellite data. And so there are a wide variety of expertise feeding into this stuff. But the point here is we have a whole bunch of protein sequences, and what we want, but we can only put in a few into a vaccine, right? You say what people want is one protein. Give me one protein to put in. We say, well, you can't get enough variability in one protein. Well, how about two? We say, how about four? And then we settle on 3. But basically, the problem is that every imaging you add, it's linear in terms of cost, and more than that in terms of risk. What's the risk that one protein won't fold correctly? Well, anyway, the point is we want to make a small number of proteins that incorporate a large amount of variability, and for this initial work, we were focused on T-cell epitopes, and there's a whole story that I won't go into now. But basically, these two things, the Mosaic Vaccine Designer and Epigraph, are tools that use different sets of different mathematical approaches to the same end, which is to put together a cocktail of a small number of proteins, which, however you slice them, will come up as natural sequences that will have high coverage of a large set of natural sequences. And again, so when you make something cool, you have to measure, well, how cool is it? And how does it compare to other options? So we have some evaluation tools as well. And so the point is here that Elizabeth showed you this map of epitopes. The key point here is this is a tiny little section of I think the GAG protein, and it's chock-a-block with epitopes, right? Everybody can present something, but they all overlap. And so you might say, well, this is a good epitope here. This, IY9 is great, but not everybody can present that. Some people, might present this thing, starting here and going to there, and a bunch of people would present that. Well, okay, you can't pick and choose and say, well, we're going to deliver this epitope and this epitope and this epitope. In fact, our approach here was to say, well, let's make the best possible version of this whole sequence and put it in there. Let the immune system sort it out. That's what it's good at, right? The immune system, the point of the immune system is to take protein sequences and present them. So the job here is not to predict the epitopes, but to present a good selection of common sequences in context, so that when they're processed, epitopes come out, and that what comes out are not only epitopes, but common epitopes. That is, ones that vaccines are likely to see that are at high frequency in the circulating population. So this is what the tool looks like. You can get there from the tools page. The point is you put in sequence and it will generate cocktails for you. The options are how many protein sequences you can get, and you know, I'd like 5, generally speaking, but the people who make vaccines say 2, so 2 is what you usually get. You can choose your epitope length. We focused on CTLs, we chose 9. You can choose, I think, up to 15. It doesn't matter very much what size you take, because they're all tiled up anyway. Point is, these things actually, surprisingly enough, and gratifyingly enough, actually work reasonably well. So we've got a number of studies for HIV and its simian equivalent. We've got studies for HIV and chlamydia, where we've shown improved immunogenicity. There's protection from challenge. So we've got some fairly nasty simian challenge study viruses, which showed excellent protection from both infection and from disease. There have been other people have used it for flu. I made one for foot and mouth disease, which has a small n but 100% efficacy. Just saying, just a little brag there. And we've got some stuff for Ebola in the pipeline too. And I've talked about this. We've got the phase 2A human trials of an HIV vaccine that are looking really good at the phase 2B. This is immunology and safety. This is efficacy trial. This is now in progress in West Africa. 2,500 women are going to be vaccinated in the near future, and we'll find out whether they're actually protected. The Mosaic tool actually uses a genetic algorithm, which is cool, but inefficient. One of the remote sensing guys, James Styler, was annoyed by these inefficiencies and rewrote this in wrote new code to solve the same problem using graph theory, and it's optimal. So the Mosaic originally gave you was a heuristic, so it gave you approximately the best answer. This gives you the best answer, and it's much faster. So you can do a lot more tweaking. And the point is, not to belabor it, if you have an alignment that you treat as a bunch of epitopes, each 9-mer, say, has a given frequency, and you can go from 9-mer to 9-mer, making different sequences. So, for example, this dashed green line corresponds to this sequence broken up into, starting as broken up into overlapping 9-mers. You can see the frequencies here. And the point is that although the variant with a C in it, cysteine, has higher frequencies here, it has lower frequencies there. And so the one with the serine ends up with better coverage. And the point is, you can make a cocktail that has both variants in, and you get good coverage. I'll skip over this part, but simply mention that there are a number of useful features here, both for building things and for evaluating them. In terms of evaluation, you can also say, well, look, you have something you want to use as a vaccine, and you have a population of target sequences. How good is your coverage? This is an important question that I think is not asked as often as it ought to be, right? What are the odds of how good a match is what you're putting in to what you're trying to get a response against? And so we have the two tools here. You add, say you put in your vaccine and you put in a target set and say, show me how good a match I have at the 9-mer level. And you'll get summaries like this, where half of the epitopes, half of the 9-mers, I should say, on average, are matched in each sequence. And if you count one by one, you're off by 1, you're up to 3/4, and so on. And you know, you get a plot. You can get a plot like this, which is not particularly informative, but if you break it out by subtype, for example, HIV is all subtypes, A through K without E, and you can say, well, in this case, your B type sequences are covered much better than your J, and so this gives you good information that you might want to address. But it's not all about a single number for what fraction of the sequence recovered. You might want to know how's your coverage by position across the gene. And again, the same deal here. You put in your vaccine, you put in your test set, and you have a bunch of things you can get out, and I'll show you some of them. So in this case, and for all the next few things, we've got the protein target going from N terminus to C terminus, and the value of coverage going up along the y-axis. So in this case, this is just a count of how many 9-mers match, or what fraction of 9-mers match each position. And this is a count of how many 9-mers are missed at each position. And if you put if you care about not only exact matches but also approximate, you get these colored ones where exact matches are red. And one intriguing thing here is the dotted lines, which will give you the sort of optimal, if you could, the best possible coverage at each position. This is sort of hard to interpret in terms of overall coverage. So if you sort these positions from best coverage to least, just take this and reorganize it, you can get a better idea of your overall coverage. And again, I forget what the overall coverage is. These are the represents the best you could do under some conditions, and this is what you actually get. Obviously, under slightly different conditions. One other thing we can do is give a two-dimensional output. So I'll walk you through this briefly. This is a sequence alignment where each amino acid is shown as a single pixel. So each row is, I'll try to go straight across. Each row, too much coffee, is one sequence, and each column is, of course, one homologous position in the alignment. So there's your sequence alignment, and if you zoom in, there's actually a fair amount of detail you can see here, even at the pixel level. There's a variable position here, and so on. Just for instance, not everyone looks at the data as much as they should, and so sometimes something that you think is aligned isn't. So I find it really handy to generate these sorts of images just from an alignment where you can see, oh yeah, this is not quite right. Classic error is to submit sequences that you think are aligned when they're not, and garbage in, garbage out. But back to coverage. The point is, if you say for each amino acid in the alignment, how many matched 9-mers is it involved in, you can get a scale here. So for example, where a vaccine matches a sequence exactly, for a stretch of 18, you'll get 9 9-mers overlapping one particular residue, and that will be in bright yellow. If you have a mismatch, something that's not in the vaccine at all, you'll get 9-mers that aren't present in the vaccine. So basically, this gives you 2D, again, which sequences are poorly matched and where they are poorly matched. So these sequences along the top, you get crap here. You're only moderately covered along here. These guys are all pretty well covered at this end, but there's some gaps over here in these sequences. It's just, again, a handy way of visualization. Just for comparison, this is the best natural isolate. So if I pick from this set the one that would be the best vaccine candidate, that's the coverage you get. If you pick the worst one, you get that. If you pick something just out of the freezer, you're liable to be somewhere in between these two. And a lot of vaccinology has been, well, what's in the freezer? Or which is to say, what grows well in culture? What's convenient to package? And that's not necessarily the best choice from a vaccinological standpoint. If you take a single sequence, a single mosaic, you can do about the same as the best natural isolate. If you add another one, you can fill in a lot of the holes that you were otherwise missing. And as the farther you take it up, the better coverage you get. Finally, I'll say the Cambr coverage you choose is not that important. The question you often get is, well, but you're optimizing for T cell epitopes. What about helper epitopes? And the answer here is helper epitopes are fine. If you optimize for 8 amino acids in your mosaic, you get this sort of coverage for 8, 9, 10, 11, and 12ers. If you optimize for 12 amino acid coverage, you drop your 8mer coverage a little bit, but it's all really fairly similar. And with that, I will say, I'm a minute late, I'm so sorry. Thanks again for being here. We'd be happy to hear from you at these addresses, both if you have questions on how you use these tools, if you want to say, boy, that was a stupid talk and you wasted my time, or or something in between would be fine. We're happy to hear from you guys. And thanks a bunch. Let's go have a drink.
On that note, also tomorrow they will have written evaluations that I'll hand out pieces of paper, so you can also put your comments in.
If you want to write written evaluations now, we can do it.
Yes.
If
They're right over there.
you
There's
have no
pens
idea.
on the other count.
If you want to write us a little smiley face, or a little frowny face. And, we'll send that into NIH in part in support of our grant.
Otherwise, the reception is going to be out on the back patio, exactly where we had lunch. You again are welcome to leave your stuff inside the room. You can bring it with you, completely up to you. And there's food out there, there's drinks out there. So yeah, enjoy.
Just for a little bit. You're right.
1103 and 2101. Just tell her reception.
One of them,
I mean, they can be
yeah.
there.
She's helping me. And I keep forgetting. It's so she goes, when are you going to send the paper? Because she's getting ready to sleep. Yeah. So I'll go home and send it to her. I just want to see what food it is.
Bad enough. You can just swallow them like that. Yes, absolutely. Yes. You may need a badge to get in from outside, but any of us can badge you in.
Well, it might be. Not gender-based. Sorry, I think so.
My daughter and I twice.
No,
No, I think it's all right.
No, yeah, excuse me. So I checked that it is that paper is all antibodies. So there's one CH505 with safety, and then it's CH103 is the antibody. So there was no T cell. Yeah. But you could do it.
Generally,
Yeah.
CTLSA, they do like, they take effector cell and like isolate PBMC
Right.
and preserve it. And then next time point when they get the like cell again, blood sample, then
Yeah.
isolate the T cell,
Yeah.
because that will be like effective effector cell, because person is also suffering from the disease, right? So and the other stored cell PBMC will be used as a like target B cell. One can isolate from that because it is from the same person. Generally, people do like this, but
Yes, there are some nice longitudinal studies of T cells. There's a paper that Mike Liu and Nilu Gunatulike. I think Mike was the first author. So it's Liu, Liu, and Gunatulike spelled just like it sounds. Am I pronouncing it right?
Nilu
Nilu.
Gunatulike.
Okay.
Yeah. But I don't know how she pronounces it. I've never met her.
and I actually wanted to ask, like in HIV, AD phenomena is there or not? Antibody dependent enhancement.
ADCC?
No,
AD, antibody dependent enhancement.
I don't think that's let's see. AD is not broadly discussed in HIV. I haven't seen it. That doesn't mean it's not. I'm not going to say it isn't present.
But naturally, it is like it's natural process. Like any virus will be like get detected by antibody, and antibody has Fc portion, and Fc receptors are there on the cell. So it
Yeah, and the question is whether it's being directed towards target cells. So the thing is, HIV can infect macrophages. Right.
That
So
macrophage
that's
that might help, like antibody role might be there.
It might.
But
it's not. But macrophages aren't the major target.
So my question is actually in relation to mosaic vaccine. So since you are like mixing several epitopes, so there will be some epitopes which might not mount as good antibody or T cell response. So those antibodies which are not neutralizing, they might be like doing
That's correct.
like mosaic.
Yeah. So the
It is Dan Baruch work.
Yeah.
Dan Baruch, right?
Yeah. So I guess all we know for sure about this in HIV context is that they do raise pretty good antibodies, and the T cell response is pretty good. Yeah. Oh, sure. The 2013 paper, the SHIV challenge was totally antibody mediated, and they got good protection. So that protection was directly correlated with the antibody response, and it had to be all envelope directed because the challenge that was the only place where the vaccine matched the challenge.
But in mosaic vaccine, they are already putting in the clinical trial. Right?
Yeah, right. Yeah. So it's the 2A study was published in July in the Lancet.
Okay.
Dan's the first author. And yeah, now so that's all done with Jensen. And it's I was going to say. So now I haven't thought about AE in an HIV context.
Yeah, I haven't. You are read, but I just asked you like.
Yeah, no, it's a thing. I mean, so for we were thinking about making a dengue vaccine, mosaic dengue vaccine.
In dengue,
some
it
years
is like.
ago. And we
big
didn't.
deal.
We looked at it, and we looked at the problem of ADE, or what I'm not even sure we knew that was what it was at the time. But we looked at the because this was 10 years ago, and I don't think the mechanism was clear.
No, it's not clear yet.
Yeah, but that. Well, but at least we have some idea, right? Now we know for sure that it's non-neutralizing antibodies that increase macrophage uptake.
But it is like in vitro. In vivo, it has not been shown yet.
Okay, fair
In
enough.
vivo,
But that seems plausible. I mean, but in any case, we knew that because because the likelihood of the hemorrhagic syndrome on reinfection was such a problem, we decided not to pursue a mosaic dengue vaccine because of the risk of.
It's very one developed like vaccine against serotype 2, but there
That's
will
fine.
be
You
like
can protect
cross
against
reactivity
serotype
for 1, 3, 4, and
Yeah.
but it will be like less immunogenic. So antibodies develop may not be like neutralizing.
Yeah.
Later, if the person get infected by the other serotype, it will do
They're in
like
trouble.
huge damage, severe disease.
Yeah, and so you really have to, and I was hopeful about the tetravalent vaccine, but apparently that's not.
Actually I heard that means NIS, right? Or Sanofi or
I think
Sanofi,
I
I
think
think.
Sanofi, but I'm not sure.
Yeah, so it is like they have some like vaccinated people should not take or something like that. Means if there is condition like either vaccinated people should take or not. Means there is some issue there with that Sanofi. It's not successful.
Yeah, yeah, it's a.
There is issue.
Yeah, I suspect it's going to be about that the solution will be getting getting broad responses targeting the correct epitopes.
Correct. So people are trying to do like ED3 domain of dengue envelope protein.
Yeah.
So all there people are making like tetravalent. So they are making ED3 all from all 4 serotypes, taking ED3 and making viral like particles, something like that, and trying to do that.
And that's fine, except the problem. You need to have a scaffold to present the protein in the right orientation.
Yeah.
And then you get antibodies to the scaffold. Yes.
So HCV was taken as a HCV or hepatitis. I think HCV, hepatitis A, was taken as a envelope of a HCV or hepatitis A, I think, was taken as a scaffold for these AD3. It is Dr. Naveen Khanna from ICG, our institute. He is trying, but still it is like study phase.
Yeah, well,
He is trying to develop bio-like particles.
Yeah, it's a difficult problem.
Dengue is a big problem. Like in India, it's still a big
I was
problem.
telling them this morning, my brother went home to India last year and got dengue. And when I was a child, nobody got dengue in India. I never heard of anybody getting that.
So
I must be
actually,
very
yes.
old, but still,
So,
nobody got it.
like 20 years back,
Now-
India was not on the dengue map,
dengue map.
and now it is like
It's very hard.
it's up.
That's what I used to hear about dengue as being a disease in the tropical Indonesian forest and in Africa. Well, that's how we were when I was a child.
Yeah.
But now, like my brother went and he was in hospital.
Yeah, so now dengue is hot spot in India and
Yeah.
that kind of climate. Because of population urbanization, several reasons.
Yeah, there's a lot of reasons, but you can tell that the epidemiology has changed and the virus
And very fast,
subtypes.
changing
There's a
very
different
fast.
subtype than the East Asia one or the African
Well,
one.
that makes you feel better, doesn't it?
We made our own. Yeah.
no.
So if you get immunized again, you can't visit East Asia and you can't visit Africa unless you pick up those.
Right.
And then die.
Huh.